Welcome to The Forest Garden, a podcast for gardeners who want to upgrade their landscapes into biodiverse food forest systems. On today's episode of The Forest Garden, we have the pleasure of talking to Dr. Sandra Anagnostakis, who for much of her life has been one of the lead researchers in the Department of Plant Pathology and Ecology at the Connecticut Agricultural Experiment Station. Listen in today as we dive into Dr. Sandy's decades worth of research into treatment of the American chestnut blight, cross-species chestnut breeding projects, and much more. Stick with us. So, Dr. Sandy, for our listeners who maybe don't know you or your research, why don't we start off by just having you introduce yourself and your background, or I guess we should just, go to where it all started.
Okay, I started working at the Connecticut Agricultural Experiment Station, which is in New Haven, Connecticut, in 1966. And my job at that time was to work on fungi that cause plant diseases because my background was in mycology. And I did that for a few years. Worked on a corn pathogen and a tomato pathogen. And then one of my colleagues in the genetics department was a former student of Arthur Graves, who had started the Chestnut breeding project. And Richard Janes came into my office and said, you work on fungi that cause plant diseases. Here's a plant disease. And he gave me a branch of chestnut, which had chestnut blight disease caused by a fungus. And he said, why don't you work on this? Well, I told him that this had all been done already in the 20s and 30s, and I didn't think there was anything to do. But because I of course, as someone who had done a lot of work with fungi, I was curious, and I made some cultures from this fungus. And I must say, it is a beautiful fungus, grows well in culture, and it was really interesting. So I started looking at some of the properties of this fungus and trying to view it from a genetic standpoint. And I discovered in looking at the older literature that there were a lot of things that had not been done with this fungus. So I started doing basic genetic studies with this fungus with my colleague, John Puhala, another geneticist. And we published a whole bunch of papers on the interesting properties of this fungus. And Richard Janes came into one of our department meetings and said, I've just found this paper from an Italian meeting where the people claim that the chestnuts, which have had blight in Italy, were recovering. And nobody thought very much about it, but a Frenchman who was there had taken samples, taken them back to his laboratory, and he was able to demonstrate that the fungus that wasn't killing trees but was clearly growing in the chestnuts, it looked different. And if he put pieces of that fungus around the edge of a killing canker on a chestnut tree, the canker stopped expanding and the tree continued to grow. So we were really excited about this because my colleagues and I had been working on a virus in the corn smut fungus, and we knew a lot about isolating viruses from fungi. So I wrote to this Frenchman, whose name is Jean Grant. He was in Clermont-Ferrand, and I asked him for some cultures that he had isolated from the Italian samples. We grew it in culture. And of course, the first thing we did was test it for viruses. And sure enough, this unusual looking fungus actually had a virus. If we plated out spores from that fungus, some of them turned out to be normal and ultimately capable of killing trees. Some of them were the non-lethal kind. So we decided to call this phenomenon hypovirulence because the virus was clearly keeping the fungus from being virulent as a pathogen on chestnut trees. So here's another whole wonderful thing to start studying. And we could study the genetics not only of the fungus now, but of the virus. And that fit very well with previous work at the experiment station because the whole idea of breeding chestnut trees for resistance to chestnut blight was begun in 1930 by a scientist in New York, but had property in Hamden, Connecticut. And he started importing chestnut trees and wanted to make crosses. So he contacted the experiment station and began working with Donald Jones, who was a very famous geneticist responsible for the whole idea of making hybrid corn. And Jones liked chestnut trees and collaborated with Arthur Graves, and they began this breeding project, which had continued up until this time. Now, Richard Janes, my colleague, who had brought all of this information to me, had been a student of Graves and Jones's when he was at Yale. So he was able to provide us with seedling chestnut trees that we could grow in a protected greenhouse and test our ideas about these new hypovirulent strains that might cure chestnut blight. We did a lot of work. Everyone in the genetics department did a lot of work on this project. We published several papers, a really nice one in Science, and it became kind of the focus of my work, because we thought that if this abnormal hypovirulent fungus could stop the blight fungus from killing trees, maybe we could save the chestnut. So we did a lot of tests, both at our farm in Hamden, Connecticut, and then ultimately some American chestnuts in the woodlands around southern Connecticut. I can tell you that the experiments at the farm were wonderful. We planted, Dick Janes planted American chestnuts. That was in 1976. By 1978, a lot of them had chestnut blight. So we started putting bits of hypovirulent blight fungus, virus-containing fungus, around the edges of those cankers, and the cankers stopped expanding. Now, what was really interesting to me in this little, relatively small study was that a lot of the trees just went ahead and died. About a third of them died back to the ground and then sprouted, because chestnuts just keep sprouting from the root collar, from the base of the tree. And the sprouts continued to grow. And the remaining group were the original trees, the original stems, and they are now very, very large trees. We can continue to put in hypovirulent strains of the fungus, around the cankers that we found. Nancy De Palma actually did most of the actual work. She was Dick James's technician. And I grew the stuff up in the laboratory and she put it in the trees. The resulting small group of 70 trees are still there at Lockwood Farm in Hamden. And the state champion American chestnut tree is one of those trees because it's huge and still doing very well. One of my questions was, would this work in the forest? So we did a bunch of experiments in the forest. We found that when the trees, American chestnuts in the forest, did not respond as well to the use of this biocontrol as that isolated plot at the farm. Our conclusion was that there was so much competition in the forest for nutrients and for water and for sunlight that the trees didn't have the strength to propel us, which walls off the fungus in a hypovirulent canker. I started worrying about what was the reason that this fungus was killing trees. So I worked with a colleague in chemistry. She had a lot of experience with pathways that were important in various plant community activities. And one of those pathways, chemical pathways that she worked on, led eventually to the production of oxalic acid. So we studied the blight fungus, and we found that the killing strains of the fungus produced huge amounts of oxalic acid, and the hypovirulent virus infecting strains produced very little oxalic acid. Evelyn and I decided that it was likely that the oxalic acid would take the calcium molecules out of the bonds between cells and chestnut bark. The fungus actually infects a tree by getting into a wound. So the fungus wound on dead tissue makes oxalic acid. The oxalic acid captures calcium molecules from the bonds between cells, and then the fungus can grow between the cells, produce more oxalic acid, which kills the cells. The fungus lives on the cell material, makes more oxalic acid, and keeps growing to eventually grow all the way around the trunk of the tree or the branch and totally kill all of the cambium, which is the living part of a ring porous tree. Chestnuts are ring porous. That means that at the end of the season, all of the vessels that are carrying nutrients from the top to the roots and water from the roots up to the leaves, those vessels get clogged up at the end of the year. In the spring, the cambium, which is on the outside of the branch or trunk, the cambium makes a whole new set of vessels to carry water up quickly to the top to wake up the trees. If the cambium is killed, no new vessels are produced, and the tree dies very quickly. There's no way to get water from the roots up to the top of the tree, or nutrients from the top down to the roots. So Evelyn and I suggested that this ability of the blight fungus to make oxalic acid was extremely important in the ability of the fungus to kill trees. And that has taken off and gone a whole lot of other directions, as We can talk about that in addition. But as far as my work, Dick Janes retired. So there was nobody left in genetics who was continuing the breeding project, crossing chestnut trees to produce new trees, looking for those which had superior timber form or superior nut production. They're very different properties. And the experiment station had this huge resource of trees that had been produced since the early, since 1930, when the first crosses were made. And there was nobody working on them. So I figured, you do genetics with fungi, you do genetics with trees, couldn't be that different. So I took on the breeding of chestnut trees, and I really loved it because it meant that I got to work outside, and it was very interesting. So I worked on breeding chestnut trees. I still did some work with the fungus, but most of my work since 1983, when he retired, most of my work has been on tree breeding.
Great. That was a wonderful summary and history. I learned a lot, took a lot from that. I think one question on some of the content you just covered that I had was, now you said, at Lockwood you were seeing, or at the experimental station, you were seeing, a certain percentage of trees responding well to the hypervirulent strain of fungus. So would that mean that there's an interaction between the genetics of the host tree and the ability for that hypervirulent fungus to grow and provide those protective effects? And if that's the case, is that where, when you find a chestnut tree that might not have, as much damage on it in its cambium and its bark from the blight, is that because of can you assume that's because of the hypervirulence, or is it possible that it's also from just the genetics of the tree itself being able to protect from the blight?
I think it has to be both. But what I continually tell people, go look at that little grove of 70 trees. That proves the difference in those trees' reaction. Some of them produce vast amounts of callus. Some of them produce very little. The ones that are not producing much callus, which walls off the fungus, those are the ones that die. So clearly there is a genetic factor in the tree which is protecting it from not necessarily from chestnut blight, but any other pathogen that should come in. It's something in the tree's genetic makeup that allows it to produce wound tissue, abundant amounts of callus, and some trees have it and some don't. It's clear that among all of the species of chestnut, and I always say there are 7 species, but I'm beginning to think there are probably more than that. But anyway, there are many chestnut trees that are pretty good at resisting the chestnut blight fungus. I would prefer not to call them resistant. I think they're basically tolerant, because one of my favorite Asian chestnut trees is a Japanese chestnut that was brought in the first importation of Asian chestnuts into the United States in 1875. That importation was an accident. At that time, there had not presumably been importations of chestnuts from Asia, and Japan had been closed off to Western scientists, botanists, for a very long time. And so when Japan was opened up forcibly, and it was possible for people, botanists, to go there and make collections and send things back to the United States, one of the things that started being brought into nurseries, and in particular to an importer in New York, there were baskets of lily bulbs. These were wicker baskets packed with sphagnum moss, and the lily bulbs were inside the moss. But one of those baskets that came in didn't have lily bulbs. It had Japanese chestnuts, which are very large, probably about the same size as the lily bulbs. And the importer in New York, Parsons, decided that he didn't really want to do anything with them. So he gave them to one of his colleagues who lived in Connecticut, and they were planted on this man's farm in Stamford, I believe. And they grew and were very different from American chestnuts or European chestnuts. We had abundant trees of both American and European in this country, of course. And the Japanese chestnuts started making nuts in a couple of years, unlike American chestnuts, which take 10 to 15 years to start producing nuts. And European chestnuts are also fairly slow. So everybody wanted one of these trees. And they were planted, distributed to friends, I assume, of this landowner. And I found one of them in Old Lyme, Connecticut, one of the first ones I discovered. And it was clearly the information that it had come from an 1875 tree had been handed down. And this is a wonderful tree. It's short and spreading, doesn't look at all like an American chestnut, which is tall and just heads straight up. But the tree was covered with chestnut blight. It had cankers all over it, and there was massive amounts of callus on the branches and the trunk. But the tree was just fine and continued to grow and produce lots of nuts every year. So this was the indication that the ability to form masses of callus is important in resisting the fungus. We looked around for others. We found other I think it was 1876, found other 1876 Japanese trees, and everything we found had similar qualities. They were not resistant to blight, they were tolerant of blight. And that was also true of the Chinese chestnuts that Arthur Graves had planted in his plantation, the first plantings that he started in 1930. And the trees, the Chinese chestnut trees also were able to produce a lot of callus, but they still became infected. So they're tolerant, they're not resistant. And that's an important difference. It's easy for people to say, oh, I found a chestnut in the woods and it's absolutely clean. It has no blight cankers, so it must be resistant. So then I would be much more interested if that tree looked like an American chestnut and was covered in cankers, but had lots of callus. That would prove to me that it was tolerant. If it has no cankers at all, it probably has just escaped being wounded and infected.
Now, that was very interesting. And Ben and I have been to Lockwood together and seen the survivor chestnut area. And what's really incredible about some of those trees, like you said, there's the state champion there, and there's lots of these trees that are just enormous, and they still produce. They produce having not you around to sort of take care of them. I don't know. I don't think anyone's working on them, or maybe, they've just been getting less attention, but they're still kicking, they're still doing great, and they're still producing a crop. And you can see where the dieback happens and whatnot. But that distinction is really important, I think, because frequently people will go out and say, oh, we found these American chestnuts that are untouched in Ohio or the Pacific Northwest or whatever. But is it that they just have luckily escaped the fungus, or is there something greater there? That's a really important distinction.
So since we're talking a bit about locations of American chestnut trees and throughout New England and the Northeast, I sort of have I guess, a more personal question for myself. Something that really hits close to home, literally for me, is I grew up in Rocky Hill, Connecticut.
Oh.
And my understanding is there were some stands of American chestnut, or maybe there still are, in Rocky Hill. And I'm wondering, like, are they still there, or did they contribute to any of the genetics, or were grafted out of my hometown into some of these other repositories?
Well, that's very interesting because Rocky Hill is dear to my heart. One of the first big experiments that I did in a woodlot was in Rocky Hill on Route 3, if you know the area, right across from a big blue gas tank on Rocky Hill, very near the southern edge of the town. There's this woodlot. It had a lot of American chestnut trees. It was reported to me by a citizen, this little plot of land, a few acres. It had never been clear cut. They had simply used the trees there for wood. They'd go out and harvest a single tree or two or three trees for firewood. That meant that the diversity in that woodlot was amazing. There were a lot of different species of trees. There were many things in the understory. It was a very good example of a typical forest that had not been very much disturbed. So my technician, Pam Slitton, and I started working on these trees. First, we mapped all of the American chestnuts. We had clearly come into this plot at a time that the American chestnuts had all died back and sprouted, because they keep doing that. They die back to the ground from chestnut blight, and then they sprout, and the sprouts grow sometimes for 10 or 15 years. And then they get wounded, then they get blight, and then they die again. So we found this woodlot at a time when there were lots of very nice American chestnuts that were 3 to 5 inches in diameter, which were growing well. And there were not very many cankers on those trees. And there certainly were some. So we took samples of the fungus that we found there. We put the virus into all of those strains of the fungus. We went back to the woodlot and we used these new hypovirulent strains to treat every canker we could reach on those trees in the Rocky Hill woodlot. We did that for several years in a row and kept track of whether there were new cankers, whether the virus spread all by itself, which of course it did. And it spread to every new canker. And we assumed that that was because every creature that walked up and down the tree, every chickadee, every carpenter ant, every squirrel, everything that went up and down the tree could carry spores of the hypovirulent virus-containing fungus up to the new cankers. And that would allow the virus to move from the hypovirulent spores into the new canker, and the canker would stop expanding, the tree would make callus. We were really excited for the first couple of years, and we were keeping track of some of the genetics of the fungus that we found. And there were lots of different strains of the blight fungus. There were genetic markers that we could use to test this. But unfortunately, after several years, the ability of the trees to compete in that woodlot was very severely limited. And they had to produce callus and fight the blight. But if there wasn't very much water, or they were competing for nutrients, they couldn't make enough callus to keep themselves alive. And eventually, all of our test trees died back to the ground. And of course, they sprouted and started again. I had vast amounts of data by this time. Now we're up to the late 1980s. And I had all this data on the Rocky Hill woodlot. And I wrote a couple of papers about it. And a visiting scientist from Germany said, you know, you could write this up as a dissertation and get your doctorate at my university. I decided to go to the Justus Liebig Institute, which is near Frankfurt, where they were famous for agricultural research. I wrote up all of this Rocky Hill data and wrote it up as a doctorate dissertation. And I ended up with a doctorate in agronomy from Justus Liebig, came back to the United States, and continued to work on chestnuts, doing more and more work on the breeding of the trees. Nobody ever asked me to see my diploma.
So you could have stayed Dr. Sandy with a master's degree if you wanted to. They would have never.
I could not have done it in good faith.
Of course. Wow, that was an incredible story. I wasn't expecting to go from Rocky Hill to Connecticut over to Germany and back.
Yeah, that's Rocky Hill trees. The Rocky Hill trees are still there. I stop and say hello to them every time I'm coming through that area. And I have a planting of seedlings from individual Rocky Hill trees, the experiment station at Lockwood Farm.
Dr. Sandy, I also grew up so Ben lived in Rocky Hill, but I grew up in Cromwell, literally a mile, my street is right off of Route 3. And my entire childhood, I had no idea that woodlot was there until I was, last year or maybe 2 years ago. I found out about it and I was like, what? And I immediately drove there and checked out all the chestnut trees. When I got there, I was maybe a little bit disappointed to see that they had all died back. That's such a diverse wetland kind of spot where there's beech and there's all these other species that are competing. So that was really valuable for me to hear about the history of that location.
Yes, that's a great woodlot. And what's interesting to me now, we have these half sieves. That is, okay, we collect nuts from a tree. We don't know where the pollen came from, but we know who the mother is. So those trees in Rocky Hill all had numbers. Tree number 5, for instance, we had a lot of seed from tree number 5. We didn't know who the daddy was, but we knew the tree number 5 was the mother. So my planting at Lockwood Farm is identified by the number of the mother tree. And I've been watching that planting for a long time now. Obviously, we've put hypovirulent strains into the cankers in that planting. And some of the offspring, some of those half siblings are much better at resisting blight than others. And yet again, here's an example that the genetics of the tree really matter in its ability not only to compete, but to tolerate the fungus if, in the case of American chestnuts, there is the additional help of the biocontrol by the virus.
Well, that's all really, really interesting. Thank you so much for this deep dive. I am really interested in some of the hybrid chestnuts and the the cultivars that have gone cross back and forth, and like the history of some of the cultivars that were named at Lockwood Farm. So, there's Lockwood, which I've seen in person and is just a magnificent specimen tree. And then Ben and I also visited the Arthur Graves plantation to see the mother tree of Sleeping Giant. And just, I don't know, these trees just blow me away in person. I'm wondering if there's any cultivars out there that are your personal favorites, or ones that you could recommend to people who are listening who might be, interested in starting some sort of plantation for nut production, or just young growers, people who are in their 20s and 30s who are just starting to plant trees on a property and, want to have a corn tree of the North for long-term generational food security.
Well, first of all, it's important to realize that there's a big difference between a timber tree and an orchard tree. A timber tree, like American chestnut, puts all of its energy into vertical growth, grows right straight up until it reaches the canopy. And when it's in the canopy and the leaves are getting full sun, then it starts to produce nuts. Because the nuts are not very important, they're very small. The nuts from American chestnuts are quite small. The Asian chestnuts generally have been bred for several thousand years to be short and spreading and have big nuts, because the people who were growing them were interested in producing nuts for food, for both humans and animals. And that's also true of European chestnuts. There are European chestnuts in the native range, which is in the Caucasus Mountains between the Black and the Caspian Sea. Those European chestnuts are very tall and excellent upright growth. But the chestnuts that have been selected and planted west of that area, that are probably European, are shorter and more spreading and have larger nuts. And there are other characteristics that have made me doubt that they're pure European, but that's beside the point. So if you want a timber tree, you go for something with good apical dominance. And if you want an orchard tree to provide nuts, you want something that's shorter and spreading, which produces large, good quality nuts, and which starts producing those nuts early in its life. I mentioned that those first imported Japanese chestnuts fruited early in their lives, and everybody was amazed and excited. So the hybrids that have been produced both on the West Coast and subsequently on the East Coast have been aiming primarily for orchard trees that produce good nuts. The hybrids that were produced on the West Coast are mostly European by Japanese. They grow very well in that climate and are well adapted to the soils and the weather conditions. On the East Coast, there were both Japanese and Chinese trees imported and used to make hybrids. There were various opinions about which Asian species were best. They were used, obviously, for getting trees with not only blight tolerance, but trees that had good orchard quality nuts. I was swayed by the work of Meter in New Hampshire, who said that in his studies, hybrids with Japanese chestnut did better than hybrids with Chinese chestnut in the climate of New Hampshire. So I started paying more attention to all of our various hybrids, starting with Arthur Graves right through Richard Jane's and looking for those which had good quality nuts and were able to survive well in Connecticut. And so my breeding efforts for orchard trees initially was to look for not only blight and good nut quality, but good survivability in the few years when we had -20 in the winter and perhaps little watering in the summers. The way my breeding program then evolved was that obviously new pathogens and new pests keep coming into the United States. And I was worried about some of these new things that were coming in. I went to visit my friend Jerry Payne, a very well-known entomologist in Georgia. And he had been the person who discovered Asian chestnut gall wasp in the United States. He found it because a grower in Georgia had gone to Japan and found some chestnuts he thought were really nice. So he put some cuttings in his pocket and brought them home, did not go through plant quarantine. He grafted them in his orchard, and eventually his orchard was pretty much devastated by Asian chestnut gall wasp, which came in the buds of those cuttings that he had brought back. Now, the gall wasp laid its eggs in the developing leaf and flower buds on the trees. That particular orchard was all Chinese chestnut, and they were all very susceptible to infestation by this insect. Jerry Payne looked at the whole situation, studied the insect, went to Japan, brought back a whole bunch of parasites that he found there and released them in Georgia, continued to study which ones were moderately successful in at least controlling some of the damage by gall wasp. The reason the gall wasp is causing damage on trees is that once the bud is infested by gall wasp, the gall wasp lays its eggs. The eggs are laid in the fall. The eggs hatch in the spring, and instead of that bud producing a leaf or a flower, it produces a gall, which is a swollen tissue with plant tissue infested and influenced by chemicals produced by the wasp larva. When the larva has grown big enough, it changes into a wasp, tunnels out and flies away to look for another tree. The leaf bud or the flower bud results in a dead leaf or a dead flower. So if many of the leaves on the tree turn out to be dead because there are galls at their base, the tree is severely stressed. And I was really worried about gall wasp getting to the rest of the range of chestnut or the places where people were planting them for the nuts. So I went with Jerry Payne through this important planting in Georgia called Callaway Farm. This is a place where the owner had planted imported chestnuts of many species in large blocks, and it was a fascinating place. And as we were going through, there were all these trees with a lot of gall wasp damage, lots of dead leaves, lots of galls. I came to an area where there were some trees that looked like Japanese chestnuts, they had no galls. I was so excited, and I was running around looking at these trees and saying, Jerry, look, these Japanese chestnuts are resistant to gall wasp. And Jerry, who managed a very slow-spoken Southern accent, said, those are not Japanese chestnuts. Those are Castanea henryi. Okay, here's another species. This species, which is native to China, is the Chinese chinkapin. So why is Castanea henryi resistant to gall wasp? The difference between a chinkapin and something we call a chestnut is that chestnuts have 3 nuts in each burr, and chinkapins have 1 nut in each burr. And the interesting thing here is that in China, there is 1 chinkapin, Castanea henryi. In the United States, there are several species that are chinkapins. We have named at least 4, and I'm convinced there are even more in the southern part of the United States. So Castanea henryi is resistant to gall wasp. Naturally, I rushed back to Connecticut and started looking at our trees. We had examples of all of the species of Castanea planted in Connecticut through the efforts of the experiment station. And I was encouraged by this, that we had the potential for finding resistance to gall wasp. Well, gall wasp naturally, as invasive insects do, made its way to Connecticut. I found the first visual evidence of galls in 2011. And I started going through our plantings, looking at the trees. Almost all of the trees, all of the hybrids had galls, but none of the chinkapins were badly infested. There might be 1 or 2 galls here or there, but none of the chinkapins were badly damaged by gall wasp. So immediately, I decided, okay, this is the next crossing project. I chose 2 commercial chestnuts, the cultivar Colossal and the cultivar Eaton. And I crossed them first with Castanea henryi, the Chinese chinkapin, planted out the seedlings. And the following year, I made crosses of the same 2 cultivars with Ozark chinkapins, because I like Ozark chinkapins a lot. The other chinkapins at the experiment station are sitting there in Lockwood Farm and also at the plantation by Sleeping Giant, just waiting for me to do some more crosses. We've examined those hybrids, and in fact, the hybrids with Chinese chinkapin, Castanea henryi, look pretty good. They don't have very good blight resistance, some of them. The cultivar Colossal is actually quite susceptible to blight. So those hybrids, a lot of them got blight and died in spite of hypovirulence in the orchard. The hybrids with cultivar Eaton did better. And that proved to be the case also with the hybrids that I made with Ozark chinkapins. The hybrids with Eaton look better, but the other thing about these hybrids is that they get galls. They're not heavily infested, but they do get galls. But instead of the tissue around the gall dying, the initial leaf that would be there dies. But then the tree makes a sprout. And so a new stem is produced, or from around the gall tissue, and the tree goes right on growing, produces flowers and fruits, and is obviously tolerant of the infest, which is less than on, for instance, on European chestnuts are just awfully, very, very much susceptible to gall wasp infestation. This is a really strange reaction to the infestation by this insect, that the tree has found a way to grow around it and has produced some hybrids that are producing pretty good nuts. And in fact, my friend Buzz Ferber, who runs Perfect Circle Farm in Vermont, has one that is his absolute favorite, which is eaten by Ozarkensis, and he says he's going to name it Dr. Sandy. So
Wow.
A new cultivar.
That sounds great. I'd love to try one of those. The Ozarkensis before, and you said you like them, and I can see why. They've got a great flavor, very sweet. And so if the hybrids have, maybe larger nuts or really great production of something that tastes just as good, I mean,
I thought so too. Yes, Ozarkensis, well, both Ozarkensis and Pumala, which is the Allegheny chinkapin. Another one of my side projects, we did some studies begun by a chemist, actually, who was interested in the properties of the various species of chestnuts, the nuts. Let me start again. The properties of the nuts from various species of chestnut. And everyone has noticed that the nuts from chinkapins taste sweeter than nuts from other species. In general, people say American chestnut nuts are sweeter than Asians or Europeans. Well, what this chemist found was that the sweetness is not due to more sugar in those nuts, in the chinkapins or the American chestnuts. It appears to be due to the presence of a chemical called oleic acid, which is found in very large amounts in the chinkapins, both Allegheny and Ozarkensis, in somewhat smaller amounts in American chestnut. So I did a bunch of studies trying to look at hybrids and see whether they were higher in oleic acid and whether they tasted better and so forth. And that I think that's somebody else's life work. I made a start, but I was interested in producing hybrids that would grow well in Connecticut or in the Northeast in general, and be resistant to diseases and pests. And I think this is a value of those plantings that people sometimes overlook. Say a new disease comes in, somebody brings something in from Azerbaijan or something like that, and a new disease, chestnuts. I can walk through those plantings of all those species that the experiment station has collected. And once the disease or the insect pest gets to that planting, I can see if any of the species are tolerant of it or have resistance to it. And then the next batch of crosses should include that species to find tolerance to this new pathogen, which is something you can only do if you have a large collection of all of the species possible of chestnuts.
That's all really inspiring, I have to say. I know that's maybe kind of a weird response, because we're thinking about the future where new pathogens are introduced, but it comes back to the importance of genetic repositories like Lockwood Farm.
I just really appreciated the kind of breakdown of these interspecific hybrids and the value. Not only, I think when people think of hybrid chestnuts, sometimes, or the hybridization of chestnut. They think of the American Chestnut Project to try to save the American chestnut by incorporating genes from other species in. And while that's true, there's also a lot of value for in taking genes from some of these other species like the chinkapins and even American chestnut and trying to incorporate that into our Chinese chestnut, or maybe I shouldn't call it Chinese chestnut. Maybe there's just the hybrid chestnut world for cultivating nuts, which I think people are very interested in. And I guess maybe that's my next question is what sort of potential do you see, I guess you can speak for the East Coast, what sort of potential do you see for scaling up chestnut production or consumption, interest, maybe just on the US at large, but I'm specifically interested in the Eastern.
Well, the Eastern is very different from the West Coast, not only in climate, but in soil type. And I have, as I said earlier, I was influenced by the work of Meter, who said that in New Hampshire, Japanese chestnuts did better than Chinese. So I've concentrated many of my hybrids using genes from Japanese chestnut. Now, in some parts of the eastern Midwest and in the southern United States, Chinese chestnuts do much better than Japanese chestnuts. They need warmer climate and a longer season. Japanese chestnuts will cope with a not such a warm climate and a shorter season. So if you're breeding hybrids to produce nuts on the East Coast, you have to keep in mind what the conditions are in your area. And there are so many young people now starting chestnut orchards. It's astounding to me. I think it's wonderful. I hope that all of those people who are interested in chestnuts will join the Northern Nut Growers Association, because the Northern Nut Growers, a group that has been going since 1910, was originally organized by hobbyists, people who were interested in nut trees as a hobby, but some of them really produced a lot of very important information about the qualities of various nut trees. And as the organization grew, the Northern Nut Growers, that is it's nut growers in the Northern Hemisphere, basically, they developed a research fund and accumulated money and started giving out research grants to people who were interested in trying things. So all of these young people who are starting new orchards can get a huge amount of information from the research projects that have been sponsored by the Northern Nut Growers. And there are still research grants available. The organization, we had our meeting early in August, and we voted to sponsor 2 new projects. The new projects are one which will look at microorganisms which make it difficult to make successful grafts. And the other project is to look at a group of cultivars, a selected group of cultivars, and test the nuts to find out which ones are the most efficient for producing flour, because chestnut flour would be a useful secondary product from the nuts. And not all chestnuts will produce a good quality flour. So those 2 projects have been funded by the Northern Nut Growers, and I'm looking forward to the results. The grafting one especially, because chestnuts are very hard to graft, and you can't reproduce a chestnut cultivar by rooting a cutting. It has to be grafted if it's going to be a clone, or therefore a cultivar. So finding out ways to keep the graft union from dying is really important. And it may seem like a small study, but that's the kind of thing that the northern nut growers wants to fund. So everybody interested in nut trees and starting new plantations should join the northern nut growers, look at all the nut growing publications from the northern nut growers, and see what's already been found out.
I'll also add too, as another plug for the northern nut growers, they have a file that you can purchase and download that has the annual reports, the research publications or pre-publications from all the way back from 19 day, and it's PDF searchable too. So if you're interested in topics, that range from many different species in different parts of the U.S. and elsewhere, there's a really great resource there, and so I'd recommend people checking that out if they're interested in that.
You can buy that tome of data by going to www.nutgrowing.org, and you can buy a full with all of that vast information. The other information that people might want to look at if they're interested in chestnuts is my webpage, which is on the Connecticut Experiment Station website. There's all kinds of stuff on my webpage, and I recommend it.
I can double recommend. I mean, Dr. Sandy, you have such the amount of resources that are on here in terms of even the USDA chestnut records of every state where chestnuts were planted throughout the past almost 100 years. It's really a resource for anybody who's really interested in this sort of stuff to check out. But Dr. Sandy, you've been so incredibly generous with your time. Thank you for joining us. And I just feel so lucky to have had the opportunity to learn. I've learned so many different things in this hour-long podcast episode. Thank you so much. And I'm sure our listeners would say the same if they could talk right now.
You're very welcome.